Hey everyone, how's it going? In today's video, we're going to be looking at packaging mechanisms of messenger molecules. So messenger molecules are produced and then they are sometimes packaged. That's why we have this question mark here. Then after they're packaged, they are eventually released and they go off and produce a response. And this response has to eventually get terminated. So in this episode, we're going to be focusing all about this packaging step. We're going to look at how messenger molecules get packaged and what enzymes are involved in these processes and more. So let's get into it. So before we get into mechanisms of packaging these molecules, we want to reiterate that a messenger molecule's polarity determines if it can be stored or if it can't be stored. So polar molecules can be stored inside cells as seen here. The reason's because polar molecules don't like fats, they're lipophobic, so they won't want to pass through the lipid cell membrane. They'll want to stay inside and so they can be stored. But non-polar molecules, they are lipophilic, meaning they like fats, so they will want to float right through the lipid cell membrane, and therefore there's no reason for them to stay inside the cell. They'll want to float right out. So as a result, non-polar molecules cannot be stored. Now, because in this episode we're focusing all about packaging mechanisms, or mechanisms in which messenger molecules are packaged or stored, we're going to focus all on polar molecules. It's only applicable to polar molecules. So now let's get into the mechanisms in which these molecules are packaged. So different messenger molecules, they can be stored in a variety of different cells. For example, some messenger molecules like histamine, right? They can be stored in mast cells. So you have histamine, but then once histamine is produced, it must somehow be transferred into this little green circle here we call the granule, and it's stored in the granule until it eventually needs to get released. Or let's say instead of a mast cell, you have a neuron. You can have messenger molecules such as dopamine, right? After dopamine is produced, it must be stored inside this green circle compartment called the vesicle and stored and packaged there until it gets released later on. So the question is, how can a messenger molecule such as histamine be packaged inside a granule? And how can a messenger molecule such as dopamine be packaged inside a vesicle? Well, lucky for us, if you're comparing a mast cell granule versus a neuron's vesicle, it's actually very similar mechanisms of packaging. So let's take a look at how these mechanisms work and what enzymes are involved. So this diagram represents a compartment that is used to store messenger molecules. So this green circle represents a storage compartment, such as, say, a vesicle or a granule or any other type of storage compartment in any other type of cell. And as you can see, this storage compartment has different enzymes on it. So let's take a look at this enzyme over here on the right side. ATPase is an enzyme, and what it does is it hydrolyzes ATP. So you guys have probably heard of ATP before, which is basically, it is an adenosine bound to 3 different phosphate groups. And ATP is the body's main source of energy. So what your ATPase does when it acts on ATP is it hydrolyzes it and it removes a phosphate group. So now, instead of having 3 phosphate groups, it only has 2 phosphate groups. And so, instead of ATP, it's actually ADP, because diphosphate instead of triphosphate. But the main point of this is because this releases a lot of energy. So, go over that real quick. Your ATPase is an enzyme which dephosphorylates your ATP, turning it into ADP, and the purpose of this is to release this energy. And the reason we want this energy is because you need this energy in order to pump a proton into your storage compartment. So here as you can see, your proton is going to get moved into your granule or your vesicle or whatever your storage compartment is, and now you have proton inside your storage compartment. And this can repeat again and again, so you can have lots of protons inside your storage compartments. So now your storage compartment has a lot of protons, a lot of positive charge. So after your ATPase enzyme has been hard at work and it's been pumping in all of these protons into your storage compartments, now your second enzyme can get to work. So what the second enzyme is called is an antiport, and it can exchange these positive protons for positive cationic messenger molecules that are outside the storage compartment. So for example, let's say you have positively charged dopamine outside, right? What this antipore enzyme does is it can exchange these protons, so it can cause this proton to leave, and it can cause this dopamine to enter, so it can exchange this. And in doing so, it can replace these protons inside the cell with your messenger molecules. So now you have successfully packaged your messenger molecules into your storage compartment, and that's the whole thing that we were trying to accomplish. So this first enzyme, ATPase, what it does by pumping in these protons is create something called the proton-motive force. It generates the proton-motive force. And then this proton-motive force is what is used to propel the pumping in of your messenger molecules into your storage compartments. So another quick thing to note is that your antipore enzyme over here, as you can see, I've written VMAT. VMAT is one type of antipore enzyme, but there's actually all sorts of different types. So you can have VMAT1, you can have VMAT2. This is the one that helps to package messenger molecules like histamine, norepinephrine, or dopamine. And you can also have something called VAT, which helps to transport and package acetylcholine. So as you can see, there's lots of different types of antipore enzymes, but they all work in essentially the same way, using this proton-motive force that your ATPase has created. Using that force as like a driving force, they exchange these protons for your messenger molecules, whatever they may be, and they're able to package them inside the granule or the vesicle. So that basically sums up how your packaging mechanisms work, how messenger molecules get packaged into granules or vesicles or other types of storage compartments. So your ATPase enzyme, it hydrolyzes ATP, turning it into ADP by removing a phosphate group, and in doing so, it releases energy, and this energy is used to pump protons, positively charged protons, into your granules. And that causes a buildup of protons in your granules or your vesicles or whatever storage compartment you have. And that creates your proton-motive force that we've written over here, proton-motive force. So now your antipore enzymes such as VAT, VMAT1, VMAT2, or any other types can exchange your messenger molecules by pumping out these protons and pumping in messenger molecules such as histamine, or dopamine, or norepinephrine, or acetylcholine, or others. And this is how these messenger molecules get packaged inside these storage components. So that basically concludes this episode. I hope that you were able to gain a better understanding of the mechanisms in which messenger molecules such as histamine are packaged into storage compartments such as granules or vesicles. I know this was a bit of a shorter and simpler video, but I really think that in these past couple of videos where we've been breaking down the mechanisms of termination or release or packaging are going to be really useful, as now we're going to start to go into specific signaling pathways of messenger molecules like histamine or norepinephrine or dopamine. And because we already have this background info, it's going to make it a lot faster and a lot easier to explain these pathways. So I'm looking forward to making these next few videos specifically about these cell signaling molecules, and be sure to stay tuned if these seem interesting. But until then, thank you guys so much for watching. I really appreciate all the help and support. If there's any feedback you guys have, anything you want me to make a video on or explain further in detail, please feel free to drop a comment below. I'd really appreciate any feedback. But other than that, thank you guys again for watching, and I'll see y'all next time. Take care.